What we mean is a DNA difference, a form of DNA that's associated with the form of a gene. So you could have a gene, for instance, in humans, blue eyes versus brown eyes. Same gene, but a different form of the gene. So in soybean, for instance, you could have a gene for resistance versus susceptibility. Closely associated with that in the DNA molecule is a DNA marker, one form associated with the resistance, the alternative form associated with susceptibility. So you can identify that marker and use it as a surrogate to select for resistance, if you will.
You know, I've always been amazed at the number of uses we find from soybeans. I guess this explains why when you see all that's inside the soybean, and that's what we're now able to do, look inside that soybean and see its potential to do even more. That's essentially what this is, isn't it? Right.
This is yeah, I guess maybe a relatively important question is, what is this DNA sequence? What are we talking about that we had from Dr. Stacy? And basically what it is these essentially billion chemical spelling units, if you will, of every gene in the soybean, the 20 soybean chromosomes, as well as the controlling elements related to those genes, and then other DNA between genes whose function we can't fathom, at least some of it at the moment. And that's divided into 50 million sections of these units corresponding to each of the 20 chromosome sets in soybean. So about 50 million of these chemical units in every chromosome, and there's one of those 20 chromosomes in every cell in the soybean seed or the soybean plant. Wow. Just like humans.
That is amazing, and you're talking to someone here, and myself. I barely got through science in school, okay? So you lose me in a hurry, but I am impressed by the potential here of the soybean. Now, I know that they're doing this, we mentioned at the outset, with the human genome. Also, rice and corn. Is the soybean more complicated than say, corn or rice, for instance?
Well, as we had in the introduction, the suggestion that we have over 65,000, and actually I think the release of the genome sequence indicates there are probably 66,000 genes in soybean. That's many more than in humans and in other plant species. And that's probably a result of the fact that the genome of soybean probably 10 to 15 million years ago doubled, and so essentially every gene is represented twice, and in some cases perhaps more than twice. So that's why we have this huge number of genes. Yeah, it's very complex. And that's what's so wonderful about this sequence is it allows us to unravel that complexity and use it for purposes of soybean improvement.
So as a researcher, you got to be like a kid in a candy store. You're like, wow, look what's unlocked, and I've got all this to explore now.
Well, I guess we spoke about this earlier. We started developing these genetic markers, as I said, in the early 90s. And by 1999, we had a reasonable map going with 500 markers, and we thought that was pretty good. In 2004, I believe we doubled that to 1,000. In 2007, we were up to 2,800. And with this genome sequence, in probably 3 months, we'll have 50,000 markers spread across these 20 chromosomes. So yeah, it is sort of like being a kid I mean, it's changed everything for us.
Okay, let's bring it out of the laboratory now. Let's go to Dr. Borma here and find out, as a plant breeder, all that he's finding and others are finding now in this sequencing and in the lab. Let's bring it out to plant breeding. How's it going to help?